Welcome to the Talking Precision Medicine podcast, brought to you by Genialis. I'm your host, Raphael Rosengarten, CEO and co-founder of Genialis, the RNA biomarker company. On this podcast, we discuss the future of healthcare and health technology, and how advances in data and data science are fueling the next industrial revolution. If you have not already, please subscribe to the show on Spotify, Apple Podcasts, or wherever you listen to podcasts. And for more information on Genialis and our unique approach to AI-driven precision medicine, please visit our website at www.genialis.com. That's www.genialis.com. In this episode, I chat with Dr. Sarah Hein, co-founder and CEO of March Biosciences, a Houston-based biotech developing cell therapies for T-cell malignancies. Sarah is an old friend and colleague from our days as trainees at Baylor College of Medicine, where we both circulated among those with ambitions of entrepreneurship. Sarah has definitely made good in that regard, as a serial entrepreneur deeply enmeshed in Houston's growing life science and health tech ecosystem. But don't take my word for it. Here's the conversation with Sarah. It's my pleasure. Today I'm here with Dr. Sarah Hein. Sarah is the CEO of March Biosciences, a really exciting young CAR T therapy company in Houston, Texas. But Sarah's going to do a much better job of telling you about March than I can. So please, Sarah, introduce yourself and tell us what you're building.
Yeah, thank you. So I'm Sarah Hein, co-founder, CEO for March Biosciences. We're a cell therapy company. Right now, our lead asset is in a phase 1 study for T-cell lymphoma and leukemia. And for those that don't know, that's just about 15% of the lymphoma and leukemic patients. Most of what you hear about are what's called B-cell lymphomas and leukemias. And the subset, it's a smaller patient population, but just incredible challenges for these patients with, unfortunately, a very, very poor prognosis and survival rate. And so the data in the phase 1 trial is incredibly exciting. We're moving that to a phase 2 study. And we're also expanding the pipeline both into additional oncology indications and also into T-cell driven autoimmunity. So really excited about what's in front of us. We've been able to move really quickly since we sort of officially launched last year when we took on our initial seed capital.
That sounds both really exciting and important. We have a really sort of broad audience here, a lot of folks who maybe come from the data science side or the health tech side. Let's zoom out and talk about biology a little bit. I was hoping you could walk us through sort of a little bit about cell therapy, kind of what are the different flavors? What is the flavor that you guys are focused on? Sort of a life sciences deep dive, but for people who maybe haven't studied this their whole lives.
Life sciences deep dive, but maybe without everybody fall asleep. Okay, yeah, I can do that. So cell therapies. A lot of times when we've thought about drugs in the past, we're thinking about a small molecule or even an antibody, and you have a drug and it hits a very specific target, and then you have some sort of effect after that. This concept now of being able to use the cells from your body as living drugs to perform work just in the way that your cells do normally, but now boosted by the engineering that we can do through medical intervention has really transformed patient care. It's changing a lot of different diseases right now, genetic diseases, long-term chronic diseases, but where I think we've seen the earliest impacts and certainly some of the most significant impacts so far has really been in oncology. The cell therapies specifically that I'm talking about are what's called a CAR T-cell therapy, a chimeric antigen receptor T-cell therapy. And all that means is we take them T-cells, these are your normal immune surveillance cells from your body, and engineer them with a specific targeting moiety. And that allows them to go after what we want versus sort of the random selection and development process that normally happens in your body. And we can also build in additional sort of boosters for those technology or for the cells to help them go further or last longer, expand more, or even be safer if needed, and then put them back into the patients. And that's what's called an autologous cell therapy. So like your cells, then go back to your cells. And so far, those have been the most potent therapies. Your body's really good at recognizing things that don't belong to it. And so we'd all love to be able to treat these living therapies as sort of the same way we treat other drugs, where you just take it off the shelf and you put it into the patient. There's a lot of people doing really, really great work In those types of cell therapies, those are called the allogeneic cell therapies. There's a few different strategies in development, but by and large, the autologous cell therapies, the first generation, so far have been the most efficacious for long-term patient responses. But we know that's our lead asset right now. We're also developing some early allogeneic approaches. And of course, a lot of people are really excited about the idea that you could create these modifications inside the patients themselves through what's called in vivo cell therapy. And so a lot of really cool places that this field is going right now. It's been changing at an incredible pace in comparison even to biotechnology as a whole, which, as you know, is developing really, really quickly.
It absolutely is a fascinating, and I mean, you can't get more cutting edge than this, in my opinion, in the life sciences. You mentioned that oncology, cancer, is sort of where these technologies and medicines have proved most useful so far. How do you, especially as a developer of the medicines, think about which cancers are most attractive to treat with, for example, an autologous CAR T? Like, what makes a particular cancer susceptible to this kind of medicine versus a small molecule or something people may be more familiar with?
Yeah. Well, I'm biased. So I'm going to say that there's, especially when you're talking about data science and some of the modern computational drug development, I think a lot of people are really optimistic that even if we've never found a single molecule to cure cancer yet, through combinations of molecules, we can come up with the right cocktails and selectively administer them to the right patients to maximize the impact. Perhaps maybe I'm a little bit more simplistic. I'm a biologist at heart. So I like to use this living machinery that we have from our cells as a filter, as our computational machine. And so I'm a really big fan of immunotherapies in general, where you can drive that natural adaptive immune system within your body. Now, where we've seen the most impact so far are in lymphomas and leukemias. Those are your hematological cancers. There's a variety of reasons, not least because it's just the most accessible to the cell therapies, right? It's all circulating together, and that's where those immune cells sort of hang out first and foremost. There is some really interesting data emerging right now with solid tumors, even where you're seeing people, again, take that sort of, let's say, biologically selected cells, right, and use what's called a tumor infiltrating lymphocyte therapy, and they take those and expand them, the ones that your body has already optimized against the tumor, but maybe boosting them through particular manipulations and then putting them back. And we're seeing some really promising results in solid tumors. And then our cell therapy is for hematological cancer, but it's been a really, really challenging cancer to go after, because if you're using good T cells to kill your bad, cancerous T cells, you have some really significant engineering challenges. You also have some potential, or historically people have had some safety effects from sort of off-tumor, on-target effects against your normal T cells. So it's a really exciting time. Now, it's been really cool, and I guess I'll just sort of say, we're really starting to see people optimize these therapies, even though a lot of times immunotherapies are thought of as more broadly applicable. There are biomarkers and additional development work that people are trying to do to predict even the right kind of immune stimulation to shift the balance of your immune system back into a productive, efficacious sort of range. And if you have this kind of cancer, which is, sort of evading the immune system through mutations A versus, a different cancer that's evading the immune system through mutations B, surface level kind of looks the same, but underneath we're starting to realize that, maybe immunotherapy A versus B is going to be more efficacious. And so there's some really interesting data coming out on that.
That's fascinating to hear, sort of a convergence of the precision medicine approach with what's a very personalized medicine approach, right? There's nothing more personal than an autologous cell therapy.
Yeah.
Cell therapy has been an idea for a long time, and it's clearly becoming more and more of a reality. What have been some of the kind of pivotal or seminal successes in the field that have sort of boosted your confidence or excitement about it? But, maybe we can also temper this a little bit by offering an example of a setback that maybe even March will be the company to overcome. But what makes it so hard?
It's, the devil's always in the details, I would say, to when you're trying to answer these kinds of questions. I think what's really exciting, what's been transformational for these cell therapies is within the last decade, we now have, especially if you're including the time that these drugs were in trial, we have drugs that can lead now to durable, some might be so bold as to say curative remissions of certain cancers. And that's just not been true. Cancer historically has always been a disease that we think of as a management sort of disease, not a disease that, even though we'd all like to cure it, historically we've not been able to cure it. And so that's been really, really exciting. The setbacks are multifactorial as we've tried to go from those initial successes to a more broad application of the power of these living therapies. Target selection, target refinement, developing the right logic gates within the cell to allow specific tumor responses to potentiate the cells in more difficult indications without overstimulating the immune system and, causing a catastrophic immune response in certain patients has caused the field some setbacks. And I would say we're actually sort of living through that right now with some of the trends that you're seeing in drug development. But I think by and large, people still recognize the power of these therapies. And so there's still a lot of activity ongoing. And through modern sort of both molecular biology, engineering, and, sort of computational methods, you're seeing people sort of start to create a much more sophisticated designs to overcome those challenges. I would point out the sort of everybody's always focused when we're thinking about tech and the merge of where the intersection of technology and biotechnology. I think we always focus on the targets and the drugs themselves, but I think the less sexy sort of logistical concerns, are substantial, particularly in these advanced therapies like, cell therapies. And you're seeing a lot of interesting efforts emerge around automation and intelligent development and rapid testing and release and regulatory compliance that I think is moving the field forward quite a bit as well.
Yeah, there's a lot that you just said that we could unpack. And I just want to say a couple of things back to you, and maybe I'll get it close to right for the sake of our listeners. You had mentioned sort of the idea of engineering logic gates, right, to control the behavior of the cells in your body, or maybe to modulate the behavior of your body once the cells are introduced. So what that means is not only do you have to figure out the right targets and engineer kind of how this cell is going to track down and find what it's trying to kill, but you also then have this synthetic biology challenge, right? When I think of engineering logic gates in a living system, this comes from the world of synthetic biology, where we've co-opted electrical engineering principles into biological design. It's an incredibly exciting field, one that I happen to love because I used to work in it for other purposes. But this does become a really major engineering problem in addition to a biology problem. And as you just alluded to, a manufacturing problem. And so it does seem like an incredibly challenging space, but again, with this notion of curing a disease, which is why we're after it. With all those different axes to kind of innovate on, but also get right, what are your ambitions for March? Tell us a little more about the company and your asset, and sort of where do you guys see yourselves differentiating and really kind of doing this different and maybe better?
Differently and better, hopefully. So our product from the start is sort of different and I would say better. So let's talk about target selectivity and that challenge that you were just speaking of. There's kind of a number of ways that you can think about tackling these problems a lot of times. When we're talking about target selectivity issues, what holds back cell therapies for, let's call it the T-cell malignancies, is really an issue of your cancer cells and your normal T cells are going to share targets. And so how do you overcome your ability to both create a product using the T cells themselves, but also preserve some of that normal T cell compartment? And a lot of people have tried to overcome this with pure engineering, where you edit out the target and maybe even generate a batch of sort of healthy normal cells as well alongside that are sort of immune to your cell therapy. And then you're trying to reinfuse all of that back into the patient. But that additional complexity, it's one of those that always sounds great on paper, but when paper meets the real world, you run into a tremendous number of logistical problems where now you're having to either make multiple products or you're trying to insert multiple constructs. When you're creating these logic gates on, say that's like biology, you'll run into real size limitations a lot of times around what you can do, and efficiencies of maybe you're avoiding size limitations by using multiple vectors to create your edits, but now you're kind of looking for intersections of multiple imperfect processes to create your final product, which creates a lot of inefficiency. And that has multiple impacts. It creates a cost issue, and it creates a a patient issue where some patients are going to have failures to get a product and they may not have time to wait on another product, right? March does this a little bit differently. Our founding scientists did a lot of work engineering up front what the right design was to leverage natural biology. It's simplistic when you get done with it, but requires a lot deeper understanding of the biology and how this sort of multifactorial computer we live with, the sort of the cell, can interact with itself to downregulate. In our case, we get rid of the target through driving natural processes, which self-eliminates the target on our good cells, but not on our bad cells. Interesting thing about that is we apply those same sort of engineering principles to the way we manufacture our products. So we create a much more streamlined product, more potent by preserving the natural cell qualities. And we're doing that without actually creating additional edits on cells. It's all in process control and selective understanding of how we can control the signaling of the cells during the manufacturing process prior to infusion into the patient. And then we've started to extend those same concepts into our pipeline development, where you'll see that sort of engineering principles first approach feed forward into the way that we think about additional product development. I sort of alluded to in oncology, but also, as we think about going into additional indications like autoimmunity, additional complexity is even more challenging when you get into generally non-lethal indications. And so it really drives additional pressure to think about how do you simplify your approaches and help them move more quickly from a patient perspective.
Got it. That sounds like some really interesting ways of combating it. So you said that you're currently in phase 1, moving to phase 2. Help me understand a little bit sort of what is the kind of timelines and the life cycle for a cellular therapy as opposed to a small molecule. And how far do you plan to go with March? Is the idea to get all the way to being a commercial biopharma, or is there a sort of a partnership future when things get really big and expensive?
Yeah, I mean, I can tell you what I think is likely, which is I think most small biotechs ultimately find that partnership of some sort with larger pharmaceutical companies is an optimal solution, whether it's through an acquisition or partnering or licensing. If nothing else, most biotech companies don't have sales forces, but you do see that change. You see them grow. It's just a very different kind of company when you're thinking about a commercial stage company versus the years of development that it takes to get a product to commercial stage, and the way you think about the kinds of people running it. So I can't predict the future, but I think balance of probability says partnering, right? So that's an easy one. How long it takes is actually pretty similar to any other molecule. What's going to drive our timelines more than the fact that it's a cell therapy versus a small molecule is really the need of these patients. And so I would not presume to speak for the future, and there's a lot of work to do between now and then. But certainly you've seen a lot of these advanced therapies in high need indications be able to leverage, say, accelerated approval pathways when they've been able to show the kinds of potent results that cell therapies promise with these longer term complete remissions of tumors. So we're optimistic that could provide an opportunity for us to take this product and get it to more patients more quickly if warranted through the data.
Hey there. I hope you're enjoying this episode of Talking Precision Medicine, powered by Genialis. Today, Dr. Sarah Hein from March Biosciences has shared fascinating insights into the transformative impact of CAR T-cell therapies. As we explore these advances in potentially curative medicines, it's clear that both precision and personalized medicine is reshaping our approach to treating cancer. If you are as captivated by these topics as we are, you'll want to catch Genialis at several upcoming events. We will be attending the BioAsia Taiwan Expo in Taipei this July, and on stage at several events in Boston this September, including the World Clinical Biomarkers and Companion Diagnostic Summit, that's September 4th to 6th, the Pancreatic Cancer Action Network annual meeting on September 14th and 15th, and the RAS Targeted Drug Development Summit on September 25th and 26th. For more information on where to meet our team, please click on the link in the description below. If you enjoy this show and would like to hear more episodes with more great guests more often, there's one thing you can do to help. Subscribe. Please follow us on your favorite podcast streaming service and leave us a review or comments. Thanks, and now back to the conversation. So you had mentioned that this is kind of I don't know if I want to say neglected, but almost an overlooked patient subgroup. It's not rare, maybe not rare, but like a medium-sized orphan population. So that has potential implications on your regulatory strategy. How else does that kind of factor into how you're thinking about building the company and building new products?
Yeah, it's not ultra rare, but it's certainly a reasonably orphaned indication. So anything under 200,000 patients in the U.S., prevalence is an orphan indication. And for T-cell malignancies, what you see generally in the U.S., about 8 to 10,000 new cases per year of these T-cell lymphomas. So certainly a far cry from what you see for even the B-cell malignancies. But when you compare the need for these patients, unfortunately, the majority of these patients will become relapsed or refractory to primary care, and there's not a tremendous number of options left for them. There's a handful of molecules available, but durability and efficacy is relatively poor. And so survival drops down to less than 20% in 3 years, right, for these patients. So that really defines the need. And so when you think about this as a market opportunity, these medium-sized orphan indications do provide a somewhat unique opportunity where it is a supportable patient population from a commercial standpoint if you can create a product that works. The reason there's not a lot of products is it's a very challenging indication. So when you're thinking about that from a commercial standpoint, although it's not the large patient numbers that you have in, say, even your larger lymphoma and leukemia populations, the amount of competition is dramatically less as well. So we think it's a very defensible patient population from a market standpoint that would provide a viable revenue source from a commercial perspective while being able to drive patient impact. And because of that need, that's why we think there's those opportunities to leverage these regulatory designations that are available for these really high need indications. So that's certainly the thesis that we've gotten behind. Unfortunately, when you're thinking about these drug development, you do have to think about sort of the economics behind it as well. And it's the thesis that our investors have gotten behind as well, is that there's this opportunity that is sizable and defensible.
That's really hopeful, and I'm always heartened to hear when there's a patient need that drives a mechanism that allows you to build something that's economically viable. It's good to see that the regulatory apparatus is trying to build alignment on that. I want to come back to some of the multifactorial decision making that someone like yourself and the CEO seat in a company like this has to make. But before we jump into that, one last question on the product and the program. It sounds to me, and maybe I'm mischaracterizing it, that the way your technology works is a bit platform-like. Like you could probably do this for multiple diseases or multiple T-cell malignancies, or even cop this to kind of have a broader pipeline still. How do you think about expanding your product pipeline? Are you doing a bunch of internal R&D, or do you have sort of a regular set of collaborators from whom you can license new potential medicines, or is it both?
I think you're always open to both, but we certainly have our own internal sort of engines through continued close relationships with our scientific leadership. And so, yeah, you're right. The initial product, that first indication is, always like to say, not the only indication. There's a few larger patient populations that are those subsets of those B-cell malignancies that we can address. They happen to express our target uniquely out of the potential targets, and you could select these T-cell indications. We also think that there's a really untapped opportunity around T-cell driven autoimmunity. And both with our lead strategies as well as some additional strategies that we're developing based off of the insights and expertise that we have safely targeting T-cell diseases that we have in continued development. How you get work done as an early stage biotech is always some combination of, collaboration with academic institutions, working with the best in the world. And then, of course, as we continue to grow, expanding some of the more functional, especially the functional development work, into our own labs. And so I think it looks like some combination of above. And I always say there's not really a right way, right? You figure out the best way to get the work that you need done, right? And then go from there.
So I want to get into getting the work done. You've talked about economics considerations. Obviously, you have to understand the biology, the science, the regulatory, there's so many things to consider. And, I met you when you were a PhD student at Baylor College of Medicine. And so how does one make that leap? How did you make that leap? What kind of sparked your entrepreneurial interest? And maybe you could just tell us a little bit about your career trajectory so far.
Yeah, I'm happy to. I think looking back, hindsight's 2020. It sounds like a much better thought out plan than it really, really was. But I'll try to be as honest as I can about some of the foibles and missteps along the way. So yeah, I started as a PhD student, trained in molecular biology and cancer biology. It was a very basic research lab. It wasn't like I was doing, highly translational work, but I did become sort of fascinated with this idea that the work that we were doing in academia, although foundational for eventual impact, and that's what you always finish every grant with, of course, and then we cured cancer, or we will cure cancer if you give us this money, right? But ultimately, commercialization is the process to me of really translating that basic research. So I became fascinated at the time from that academic perch in not academia, which I think is how most people start thinking about this. I got really plugged into the local ecosystem with people who were working on various startups, both as employees, some as founders even. Most were not doing biotech, but just really became fascinated with that yes and attitude that a lot of entrepreneurs bring to solving challenging problems. And managed to meet enough people and learned enough that I wish I could say it was perfectly planned, but it wasn't. I got lucky, just like, anything else. I always like to say it's cultivated luck, because if you kind of give yourself the opportunity to get lucky, then, certainly it can look like you planned it out. But I was able to join Mercury Fund as a venture fellow straight out of graduate school, working with Dan Watkins, and then helping launch a small startup called Curative Therapeutics, which was a retargeted cytokine company. And I was the only full-time person. I was definitely the most junior person, but I was the only full-time person in that company for quite some time, which means I got to interact with every aspect of that business because it was being run virtually, and seeing, both the decisions that we're making to continue development, as well as sort of the barriers in front of us that were stopping us from getting to the next stage. And then we slowly were able to chip away what I call the stopping questions, or the but for answering X, Y, and Z, you could raise the next block of money, you could sell this, you could do whatever. And we ended up selling that company to Vail Health. I then went over to TMC Innovation with two ideas in mind. One was that I want to take those hard-earned lessons and try and create a resource for people who wanted to do biotech development here in Houston, because we have the largest medical center in the world, 2 comprehensive cancer centers. There's a 3rd comprehensive cancer center just north of us in Dallas. And we have CPRIT, the Cancer Prevention and Research Institute of Texas, funding a tremendous amount of research. But historically, and just to be perfectly honest, we've been really poor overall at commercialization and taking that research and translating it into companies, particularly those that stay here. So that was the goal. The other goal was to have a place to interact with a lot of really smart people and maybe think about starting another company. And so I got really lucky in that first cohort. Max Van Moken and Malcolm Brenner came through the cohort, with this technology that fundamentally became the foundational science for March Biosciences.
That's a great version of the story. I also remember the early Houston ecosystem, and it was really, really wonderful fortune to get tuned in with Dan Watkins and the Mercury folks. They were, frankly, the only ones in the game probably at that time, or at least, but really thoughtful and in my opinion, folks who bat above their weight by a lot.
Absolutely. And I got to say, it was a wonderful opportunity, not just to get to launch a company as this more junior person with people who were, a lot smarter than me, a lot more experienced, but also sort of sitting in that environment, and even the deals that were coming through, not related to biotech, kind of hearing when the door closes and the entrepreneur sort of leaves the room, how are people thinking about risks and challenges and sort of vetting out internally what the opportunities are. And I think it's a little bit like, I don't think you understand how to get a job really, even if you've applied for a bunch of jobs, until you ever have to try and hire somebody. And you really hear how, like, you're thinking about it from the other side of the table, or until you review a grant, you don't really know how to write grants, I think. And it's sort of the same thing. Until you're sitting on the other side or hearing how people are talking about it, I don't think you really understand the limitations and opportunities that venture is constrained by themselves, right? Because they have to go fundraise. And so I think a lot of entrepreneurs forget that the VCs that they're pitching to also have to go fundraise. And so a no could come from, it couldn't be your technology, no, but it could also be constraints. It could be their thesis. It could be internal politics. It could be conflicts of interest or, specific interests of LPs. And there's just so many different variables that you can't get hung up on it, really.
I have just a couple more questions, but before we leave this notion of entrepreneurship and the Houston ecosystem, give me a sense, or give our listeners a sense from your eyes, where is the Houston, I want to really focus on health tech, biotech, med tech, so the stuff that would happen down at the Texas Medical Center. Where is the ecosystem in its maturation from, I don't know if we want to use dog years or people years, but is it a teenager? Is it like, how grown up has it become? I left town in 2018, followed my wife around the world a bit, and we just came back last fall. And it seems like there's a lot more going on, but you've really been building it with your own two hands. So
I think we're like adolescent, right? I think people are starting to see it come together, and there's a lot more activity than there used to be. There's a lot more optimism that you really can build companies here. And I got to say, there's a lot more talent. I think historically we've been a little bit negative on ourselves on having the right management or, drug development or med tech development or digital health sort of expertise to be able to build companies. And I've seen a whole new crop of leaders emerge within the ecosystem and build off of the early wins from the people who were already here and continue to build. And so that's starting, you can see the virtuous sort of cycle starting to happen. But we're still definitely a disconnected ecosystem, right? We don't have the deep relationships with capital. We don't have as much of the community and experience for people to be able to reach out to. As you know, you started a company, right? So, when you're starting, it's a lot of perspiration and aspiration and reaching out to people, trying to figure out if this could be a real idea or if this is something that you've kind of gotten yourself wrapped around and you're pulling on threads and just trying to find the right people to talk to help you put a little bit of structure to answer all of those questions that you were talking about. The complexity. Nobody has all the answers when they start, no matter how many times they've done this before. So I think we have to continue building that, but you're seeing it happen and you're seeing the opportunities emerge as we start to become more sophisticated. So adolescent, definitely where it would put us. A lot of room to grow still.
Awesome. The next one is a bit of just a olive branch to some of our listenership who really are coming in from the tech side of things. We talked a bit about logic gates and synthetic biology and design, but how do you see the emergence of really advanced analytics like AI, ML impacting cell therapy? Is it gaining some of the hype that you see it, really sort of flooding the field for like small molecules and protein design and so forth?
You see a lot of activity around, say, target discovery, but I think predictive analytics or sort of predictive AI from a drug development aspect around, say, small molecule design, docking, creating backbones that you never would have thought of from prior molecule backbones. That's been really interesting. Proteins, I'm seeing some really interesting work where people are merging LLM, these large language model type structures and concepts with protein design, because at the end of the day, it's its own kind of language, right? And there's sequences and structures within it and repeatable patterns, but just like language, just because there's repeatable patterns, there's also an infinite number of ways to create that combinations. And then trying to predict the function on the other side is incredibly challenging. And there's been a couple companies that have done this successfully already, but I think it's going to go from a very rare, very bespoke process that's locked away into these companies that make something and then get sold for a bunch of money so that way a big pharma company can get the algorithm as much as the drug, to something that I suspect will become a tool that everybody uses every day. When you go to do protein design, you'll do protein optimization with predictive functional outputs that then get validated again by the wet lab. I'm also seeing, again, I always like to say the tools and picks sort of thinking around drug development. I think it's really underappreciated in the press releases when you're thinking about it, but there's really cool AI happening around clinical trial design, around predictive and adaptive trial designs, around biometrics, again, feeding back into the trials to refine really quickly and take a trial that maybe historically would have been a failure, and because it's able to adapt as part of that inherent design, make it a success at the end because they were able to adaptively identify the right patient population and focus in on it. The regulatory allowances for that, I think, are still catching up, but the technology is already emerging, and it's very, very interesting where people are showing that. You're also, look, drug development, it is a regulated industry. A lot of drudgery around, documents and paperwork. And I think just like you're seeing for legal, you're seeing really interesting companies emerge to help speed up and democratize, I would say, the process of IND development and study reports and lab book compilations and those kinds of things. And so it's going to let us move a lot more quickly in the future in a very expensive and complex industry.
Yeah, no doubt. It's probably the best thesis for starting a new startup is solve the drudgery. Don't try to solve the exciting problem, solve the boring problem, right? But I'm glad you mentioned the future. Last questions. If you do your job right at March, if your colleagues, in the field get it right, what does the therapeutic landscape in oncology and beyond look like 5 years from now, 10 years from now? What's your rosy view of what's possible?
Yeah, I mean, look, I'm not going to promise to cure cancer, right, or all cancers, but I think base case we cure a cancer, right? That's I think an achievable goal, and it still feels pretty dang lofty from where I'm sitting. So we're going to go after this one indication. Loftier goals, though, is I think we have an ability to create sort of a new therapeutic class to target kinds of diseases that people haven't been able to target before, with all these T-cell indications more broadly, helping realign the immune system back to where it should be by being able to very specifically target that immune compartment. So it's an exciting time to be here. So we have sort of our base case, and then we have our even loftier goals at the end of that. So
that's wonderful. I won't hold you to it, but I'm looking forward to when you cure a cancer and then we can go cure the rest.
Actually, I think I can just be done at that point. If I actually ever get to say that and say, not only do we have an approved drug, but there's, people who get to live their rest of their lives because of that. I think I can just retire. I'll figure out the money part later, but that won't be the important aspect.
Take a trip
there.
to Stockholm first and then you can retire. Sarah Hein, thank you so much for joining me. This was really, really fun to talk about, and best of luck with March.
Thank you so much. Appreciate it.
This has been another episode of the Talking Precision Medicine podcast, brought to you by Genialis. Hopefully by now you've subscribed to the show, but if not, go ahead and click that subscribe button and leave us a review. And please do pay us a visit at www.genialis.com. That's www.genialis.com to learn more about how AI is transforming precision medicine. Thanks for listening, and see you next time.